Okay, we are almost at the end of our little talk about membranes, plasma membranes, and how things move across them. Um, we need to finish up with a more specific discussion of one particular substance and how it moves across cell membranes, and that substance is water. So that's where we will finish up on this little topic. Talking about osmosis, the movement of water across membranes. Okay. Now, water kind of is talked about and it's with its own little vocabulary and its own little chapter, so to speak, because it is so important to life. It is so ubiquitous. It's everywhere. When we're talking about life, we're talking about cells. Water's in them, water's outside of them, and water is always allowing the activities that a cell does to occur. Solutes are dissolved in the water that the cell is interacting with, and so on. So water is absolutely critical, and it's given its own special place as we talk about things that move across cell membranes. Okay? Osmosis really is simply the diffusion of water. Remember, diffusion is the movement of any substance from an area of high concentration to an area of low concentration. Well, osmosis is simply the movement of water from an area where there is a lot of water to an area where there is less, from an area of high concentration to an area of low concentration. Okay. Additionally, when we're talking about osmosis, we are usually talking about the movement of water from high to low concentration across some semi-permeable, selectively permeable membrane, a cell membrane or a plasma membrane. Now, when we are talking about the concentration of water, how many water molecules are in one area versus how many are in another, oftentimes we determine that by looking at how much solute is dissolved in two different water areas. Okay, so the amount of solute, something dissolved in the water, is really going to determine how concentrated, pure the water is. The more solute you have dissolved in water, the less water per unit volume you have. Okay, the solute is getting in the way of all the water molecules, and so if you were to select a sample of water that had a lot of solute in it, some of that water would be, displaced because there's solute in there taking up space. So there's some vocabulary we need to deal with this, and the first word of that vocabulary is up here. Hypertonic is simply talking about a sample of water in which there is a lot of solute dissolved in the water, more solute, and because there is more solute, there is less water in a given sample. Another term that we need to be aware of is hypotonic, and this would be a sample of water that has less solute in it and by default more water. So let's look down at these pictures. Okay, the blue indicates water. Okay, I've lost my pen. Here we go. The blue indicates water. The yellow dots are indicating solute that's dissolved in water. Okay? So if I've put a lot of solute over here, you can see that there's not as much pure water. I've got lots and lots of solute dissolved in that. Okay, whereas over here there's less solute, which means, as you can see, there's more pure water in between the molecules. Okay, so hypertonic means that I have lots of solute and therefore less water. Hypotonic means there is less solute and therefore more water. And when we are talking about osmosis, please remember that what you are concerned about is how much water do you have. Okay, it is the concentration of water being high versus somewhere where water is low that we are focusing on when we're talking about the movement of water due to osmosis. Okay, there is one more word called isotonic, and this one is pretty easy. In an isotonic solution, you have equal water amounts on each side. So if this membrane is here, there would be equal solute and equal amounts of water on both sides, and so there would be no difference between either side. You would be in a state of equilibrium. Okay, so if we look at this picture down here, the greater amount of water is on the hypotonic side. There is more pure water over here than there is on the hypertonic side. There is less pure water. The solute is in between all these water molecules, and so there's less pure water. So remembering that water moves from an area of high concentration to low, water is going to move from the hypotonic side to the hypertonic side. So water moves from a hypotonic or high concentration of water to a hypertonic or low concentration of water solution. Okay? Now, please remember, this is a low concentration of water here, but a high concentration of solute. So make sure, if you are given numbers, that you understand if the numbers are representing the amount of solute or the amount of water in a solution. Okay, because it'll make a difference in how you look at the problem. Alright, so water would move from the left to the right in this particular image. Okay? Less solute, more solute. Okay. Now, this movement of water can make or break a cell and determine whether it's going to live or die. Cells need to keep water balanced inside of their cell membranes, okay, as compared to the outside. And the effects of water in their environment, whether it's a freshwater environment, a balanced or isotonic environment, or a saltwater environment, is going to determine what happens to a cell. Okay, so let's consider the first situation. If you place a cell in a hypotonic environment, so what does that mean? That means I'm going to put my cell, here is a beaker of water, and I'm going to drop in my cell, and this water has less solute but more water than the cell. The cell comparatively has more solute, but therefore less water than the cell. So in that case, water moves from an area of high concentration to low. Water would flow into the cell from outside its environment. Okay? Now, so a cell that is in a freshwater environment, freshwater meaning the environment has lots of pure water, not as much solute, okay, is going to cause a cell to take on water. The water will move from the environment into the cell where there's lots more solute, dissolved substances in the cytoplasm of the cell. That will cause, unless the cell is able to do something about it, water to come in to the point where the cell is simply like a water balloon, and eventually you put too much water in it and the cell bursts. That bursting of a cell is called lysis. Okay. Now, an example of this is a freshwater organism, an amoeba, a paramecium. These are little animal-like protists. They're not animals because they're unicellular, but they're little protists that swim around in freshwater environments, and their bodies, their cells, their single-cell body that they're composed of will constantly take on water in this freshwater environment. And if they don't do something about it, their little bodies will look very much like this cell and just explode. So what these particular cells and other cells as well, one way they deal with this is that they have evolved a contractile vacuole over millions of years, and this organelle is simply a pump, if you will. It's a vacuole is a enclosed sac, but a contractile vacuole is a sac that is going to take in water and then pump it out of the cell's body. Okay, so it is obviously, if it's a pump and it's forcing water in a particular direction, ATP, or energy, is going to be required to make it work. So this is an energy-intensive operation for a cell. Now, plant cells are different, which we are seeing down here in this picture. Plant cells, as you hopefully remember from first-year biology, have cell walls made of cellulose. Okay, very stiff, very protective. And what this means is the cell wall is quite firm. And if we put a water balloon inside this wooden box, if you will, and keep pumping water in it, we can pump a lot of water in there because that wooden box is sturdy and that cell is not going to explode. That cell membrane is not going to explode. The cell membrane you are seeing right here, the water balloon, if you will, the cell membrane of the plant cell here, but then it's got this nice protective cellulose box around it. So that cell can take in an awful lot of water, and actually prefers to, because that water is what holds the plant upright. Obviously, you know, if you don't water a plant, it falls over. This is called turgor pressure. This pressure, this high pressure of water that holds a plant cell very upright and firm. And we say the plant cell is turgid if it is full of water like that. So the cell wall then becomes very important in plant cells. They do best when they have lots of water flowing into them, and it keeps them turgid and keeps the plant body as a whole very upright. Okay, so plant cells love it when they are in fresh water, and that's why the grocery stores, they'll spray water on the produce that's in the display, and that's what keeps the cells from wilting and drying out and not looking too good. They take that water in. And unfortunately, animal cells don't have a cell wall, so they will explode if they don't have a contractile vacuole to take care of the situation. This is just an image of a paramecium, and you can see the contractile vacuole. It's this structure here. It's got these radiating arms that come around, and you can see that the radiating arms take water in, and so this contractile vacuole here is full of water, and in this case it is pumped the water out and is much smaller, and again you can see the radiating arms around it. Okay, so that's the pump that would work, and again energy is required. Okay, if you, on the other hand, put a cell in hypertonic solution, meaning you put it in some salt water, it's going to have a very different response. In this case, if we put the cell in a beaker of water, here's my cell, and we put the salt water, a really salty water on the outside, and it's not that there's pure water inside a cell. It's not. It's got lots of solute in it, but it's got less solute than, say, ocean water, for example. So we've got really salty water here. That means the concentration of pure water is higher in the cell. So the concentration of water this time is higher in the cell because we have an even more solute out here in the environment. So consequently, the concentration of water is lower out here. And if that is the case, then water will move from high to low and it will leave the cell, which is not terribly good for a cell either. So, in the case of a animal cell, the animal cell will simply shrivel up. Again, it's like a balloon losing air, a water balloon losing water. It's just going to shrivel up and deflate and collapse. Not a good situation when it loses water. Okay. The plant cells, on the other hand, well, not on the other hand, they do the same thing. It's just that they have this cell wall that is around them that will kind of stay looking like a box, but it's not going to be near as well supported as if the cell membrane, which you can see here having collapsed away from the cell wall because it lost water, water went out into the hypertonic environment. The cell membrane simply collapses away from the cell wall, and the cell eventually will just not be able to support itself, and that's when we see plants wilt and their cell walls just kind of begin to crush because the water balloon inside is not kind of propping them up anymore. Now, plant cells can recover from this, but if it's not dealt with, of course they can die. In the winter time, when salt is laid down on the roads, you'll often see just brown strips of grass by the roadside because that salt has pulled all the moisture out of the plants by the side of the road, and they die, the grass plants by the side of the road. Okay? Alright, so in that case, both of them definitely need water. Alright, and of course isotonic simply means that the concentration of solute and water outside is equal to the concentration of solute and water on the inside. There's no difference. So in that case we have a state of equilibrium, and this would apply down here and up here, and the cell is reasonably happy. Though honestly, this plant cell would like to be even more turgid. It would love to have that turgor pressure keeping those cells really, really inflated. Okay, so no net movement of water. Now remember, it's not that the water molecules don't ever move when you've hit equilibrium. Of course they're moving. They have kinetic energy. It's just that there's really no difference in the amounts of movement from one side to the other. It's in a state of equilibrium. A great example are your blood cells. If you take your blood cells out of your body and put them in a beaker of distilled water, they'll explode. If you put them in a beaker of salt water, they shrivel up. If you put them in blood plasma, the fluid they are normally traveling in your body, they will be happy. And that's why when you are given fluids in the hospital, an IV fluid, it's a slightly salty IV fluid. It's not pure water. It's got solutes in it so that your cells continue to be in their happy environment that they need to be in. Okay? All right. Just make sure you recognize this term. Aquaporins are the little openings through which water molecules travel. They are protein channels. It was thought for 100 years or more that water molecules just, if you've got cell membranes, that water molecules just were so small that they could just move in between the phospholipids. But there were water movements that were observed that simply occurred too fast for that to be true. And what they discovered was eventually in 1991, and then this man, Peter, I think it's Agre, was awarded the Nobel Prize for discovering this protein channel called aquaporin that allows water to pass through cells, and it's ubiquitous. Aquaporins are found in everything, in you and me, and plants, and protists. It's really extraordinarily universal. Okay, so protein channels. Now let's look at this. This is our last slide. Do you understand osmosis? So what this is doing is this is giving you the concentration of solutes, and notice there's some different solutes here, but we don't care. We'll just consider them solutes. In the cell, okay, we've got 0.03 molar sucrose, 0.02 molar glucose, and in the environment, in the beaker, we've got 0.01 sucrose, 0.01 molar glucose, 0.01 molar fructose. Now, let's just add them up. So sucrose plus glucose, 0.03 plus 0.02 is 0.05 molar solution in here, so 0.05. And then over here we got 0.03, if we add all those together, molar solution out in the environment. Okay, so that is the concentration of solute. Okay, so now where then is the concentration of water, not solute, but water, higher? In the cell or in the beaker? Okay. The concentration of water is higher where there is less solute. So that means the concentration of water is higher out in the beaker. And the concentration of water is lower in the cell where there is more solute. So water will flow from out to in. So in the cell compared to the beaker, is the water hypertonic or hypotonic? The cell is, already did that, hypertonic. It has more solute. Beaker water compared to cell then is hypotonic. It has less solute, and so water will flow into the cell. Okay? All right, please remember it's all relative. I can't just show you a beaker of water and say, is this hypertonic or hypotonic? You would look at me, I hope, and say, well, compared to what? It is all relative. You have to compare the sample of water you have to some substance, some water, in some other location. Okay? All righty, that's it. Thanks for watching. I'll see you in class.